So, Marc, one angle of research in your lab is trying to better target cancer treatment, make it more personalized for different patients. Can you tell us more about that?
Well, yes, in previous times there were, to some extent, limited amounts of cancer drugs available, and all patients with a given cancer got the same drug. As a result, some patients did not respond at all. And as another result, many patients became quite rapidly resistant against that single treatment. So therefore, obviously, already 20 or 30 years ago, there were, to some extent, mixtures of anti-cancer drugs that were used. They were initially very efficient, essentially in childhood cancer. These combinations are very efficient. And over the last years, one could understand why these combination treatments are much more efficient than single-agent treatments. In fact, on one hand, cancer cells are addicted to selected proteins. So they overexpress proteins that prevent cell death. So the cells, it's like doping for the cancer cell. Obviously, when you hit this target with a given drug, cancer cells will initially die very rapidly. But unfortunately, in the cancer cell, there is a high level of redundancy. So one protein can replace another one. So if you hit the first target, the cancer cell will then switch and overexpress a second similar protein.
I see.
And will survive. Now, the idea would be to select an array of compounds based on their target specificity in order to inhibit then different proteins that have similar functions. The inhibitors can be of both natural or also synthetic nature. So there are very exciting molecules that are undergoing clinical phase 2 and 3 investigations, and we try, in fact, to combine additional molecules to these treatments to further improve, actually, the outcome, reduce resistance, and potentially also reduce the concentration of the individual drugs in order to further reduce resistance mechanisms. Because the resistance also comes sometimes due to high concentrations of a single compound to be used. So if you can attack the cancer cells with individual molecules, but at much lower concentrations, you might reduce the resistance onset.
Oh, that's interesting. And this sounds promising because you're hitting a number of different points that make cancer strong and difficult to fight and resistant to our efforts. That's interesting. Now, how does all of this help us make cancer treatment more personalized?
So these treatments obviously have to be adapted to progress in effect in a related research field, which would be in the field of genomics, where you would in a first step profile the different patients for their expression levels of selected target proteins. Then you would classify these patients, and then you could select the drug or the drug combinations for a given group of patients. Indeed, if the BCL2 protein is not expressed in a given type of cancer, there is no need to use an inhibitor for this compound. In another patient, this might be an important protein, so you would then rather target the BCL2 protein. So altogether, our combination treatments aim to target individual proteins that are expressed by subgroups of patients to prevent the cancer cell to switch from one pathway to the other and to survive. So we aim to hit the cancer cell at related targets with different compounds, and as such we induce a very efficient synergistic effect between the two compounds.